All right, we are recording, so let's do attendance. Van Cross. Kenzie's here in person, a little bit of extra credit. Michaela's here in person, a little bit of extra credit. Reed, Hemmings, Bowers, Asbury. Tudor's here in person and in red, so a little bit of extra credit. Felder, Hudson, Burdette is online. Hudson is online. Asbury is here. Little extra credit. David, Hodges, Eric, Midkiff is here and wearing red and in person, little extra credit. Same with Rice. Cedric, Richardson is online. Massey, Newton. Yep, we covered everybody. Let's see who shows up. It's 8. I'll wait till 8:01 to get started. I guess since we're recording, I don't think this is going to matter for any of you guys, but as a reminder, because some people still haven't done any independent work, like not one paper. And if you have not dropped in the class today, you're going to want to do some independent work. So let me remind you, because it's been a while since I said this, because for those of you who have been doing independent work, well, you've been doing it, so you know how it works. So just some reminders. There is no maximum or minimum, right? Your papers can be as short as you want. You don't have to do a page. Yes, it's currently worth 15 points a page, but officially worth 10 points a page. So even though it's per page, it doesn't have to be, right? You can write 2 sentences. You can write 10 pages. It doesn't matter. When you write independent work, it can be as long as you want, right? Obviously, if you write shorter papers, you're going to need to write more of them because shorter papers are just worth less. And that's okay. If you're like me, you don't like writing long papers. So just write short papers if you want to. Another thing, make sure you use references. You have to include your sources. Make sure you don't plagiarize. Most people don't straight up plagiarize on purpose. Like they don't just copy it and say that's theirs, usually. But one of the things I usually see people do, because they don't know it's plagiarism, so I'm reminding you now, is rewording something. So they'll take a sentence, like the paper will be the same as their source, sentence for sentence. Like each sentence says the same thing in the same order. They just change some of the words. Like they change some of the adverbs, they change some of the nouns, they change some of the adjectives, right? But they're still meaning the same thing. It's like they use the thesaurus, or maybe their brain, to just change some words that had the same meanings. Does that make sense? So don't do that. That's copying. And I'm telling you, if you're under the gun, and a lot of you are, please don't plagiarize. Just take a low grade if you have to. Please don't plagiarize. You're going to get yourself a 0. So I know it sucks that you've got a lot to do, you've got a lot to catch up on, but don't copy, don't cheat. That's going to hurt you more than anything else. And also to remind you, for independent work, it doesn't have to be papers. Like you do need to do some papers. That's where you're going to make the most points. But also you can get independent work for bringing in cans. Read the announcement for details, or for reading the announcements, right? Because a lot of the announcements, somewhere on the announcement says, if you read this, reply to the email and you'll get independent work points. So do those things. Get your points. Watch those videos, right? You can watch videos and answer questions for points. If you have other ideas of ways you can get independent work points, let me know. So yeah, that's it for the reminder. We had some people come in. So let's go through this one more time before we get started. David Hodges is here in person, so that's a little extra credit. And then Eric, right? Felder? Eric. Yep. Gotcha. With a little extra credit. And then we had, Eric. I know I saw Eric somewhere. All right. You're online too? All right. I saw it twice. I'm glad I wasn't going crazy. And then Cedric is online now. Where are we at here? Okay, Cedric. And I think that's it. Yep, we're all caught up. Another thing for independent work, if you haven't joined the Facebook group yet, do that too, because I try to post opportunities there for ways you can get extra credit / independent work. It's all the same for this course. So any questions about independent work or anything else before we get started on this back into chapter 10? All right. Now's the time. Like I said, if you are not withdrawing from the class today, which is the last day to do it, now is the time to really get to work. If you have a D or F, you've got a big mountain ahead of you, but it's possible. If you have a C, you've probably got enough inertia to just keep rolling. If you just keep doing what you're doing, if you have a C or above, you should be good. There's not going to be any kind of big surprises like one of the exams is going to be a lot harder than the other ones or anything like that. So here we go. Jumping back into it, back into chapter 10. We were talking about Watson and Crick and their discovery of the shape of DNA. Remember, we talked about everything we knew prior to that. And, again, I've mentioned this is not a history class, so the history of biology is not important for you guys. But in this case, it sort of is. Not that I'm going to ask you these dates, because this isn't trivial, right? It sounds a little bit trivial, like, oh, they figured out the shape of DNA. But remember, like I said in the last lecture, figuring out the shape, then, the form, then allowed us to figure out the function. Like, we didn't know how DNA worked. We knew what it did at that point when they were working, but we didn't know how it worked. And since figuring out the shape, and then since figuring out how it actually works, we've been able to do amazing things, which we'll get into in the next few chapters. But here we go. Anyway, let's get to the slide. In 1953, they published their paper, and then they won the Nobel Prize almost a decade later in 1962. Also, a guy named Wilkins received a piece of the prize for it. You can look him up if you want for independent work. What did Wilkins do? Also, Franklin deserved a share, but she was already dead at that point, so she couldn't have been awarded it for that reason. Also, there was other people involved. So again, good independent work topic if you want. Who else was involved in this? You could just write a paper where you just list it, and that's it, right? Just a list of names and just get a few points because it's a short paper. Or you could get into it and talk about what those people did. It's up to you. All right, so again, back to the important part, which is the shape of DNA, what they figured out. Watson and Crick wrote that the structure, this is in their words, immediately suggests a possible copying mechanism, the grief, for the genetic material. And that's what I've been getting at, right? It's not just some trivial stuff like, oh, we know the shape of DNA, how pretty. Now, every science teacher in America can put a picture of it on their wall, right? No, it's not like that. It is actually important. Before that, they did not know, well, they still didn't know technically at that point, but they didn't know how DNA copied itself. But once they figured out the shape, once they figured out the structure, then it was pretty apparent how it worked. Because the structure of the DNA points towards the molecular explanation for life's unique properties of reproduction and inheritance. So like there was a bullet point last lecture where we said something like DNA's unique identities are the things that it can do, which is, dictate the production of proteins and copy itself and get passed down from one generation to the next. And again, once we figured out the structure, that's what we figured out. So this slide is just a segue into the material that you're going to need to know. There's nothing to study on this slide. This is still an introduction. We're getting there. And where are we getting? To this point right here, this third bullet point. Matter of fact, that'll be the first attendance word. I almost forgot. So the first attendance word will be bullet point. So if you don't mind, send a picture of a bullet or a point. If you're in person and you want to send those words for extra credit, then it is, bullet point. Anyway, let's talk about, I thought you were raising your hand. Let's talk about DNA replication. Now we're getting into the really important stuff. As far as the exam is concerned, this first bullet point that we talked about yesterday was very important because it was the DNA and RNA structure, but that was a review from chapter 3. And now there's this that we're about to talk about. This will be on the exam. This is important information to understand how viruses work and how vaccines work and things like that. This is very important information. Here we go. DNA replication. As you already know, when cells reproduce, they have to duplicate the information, providing a copy to the new offspring while keeping one for themselves, right? You know that. That one bullet point is basically chapter 8, right? So there's really, you can write it down if you want, but you don't need to, because that bullet point is chapter 8. That's a short way of saying cells go through mitosis. Cells go through S phase of interphase, and then they go through mitosis, right? So they duplicate their chromosomes. So Watson and Crick's model suggests that each strand serves as a template to guide the production of another strand. So even though we're going to talk about it, so there's no need to write it down yet if you don't want to, but as far as the exam is concerned, the most important thing on this slide is this. Each strand of DNA serves as a template to guide the reproduction of another strand. And that'll make sense when I show you how it works. Matter of fact, yeah, so in a sense, I guess that's not even that important either, because you don't need to memorize this fact, but I'm going to show you how it works. And that's what you need to understand, is how it works. You need to know how each strand is going to serve as a guide to reproduce the other strand. So any questions about this slide? Okay, moving forward. Here's what I'm talking about. Here's the original up top, that blue part. That's the original DNA double helix. And as you know, again, to remind you, because I haven't said this since Wednesday, when we look at a double helix of DNA, that's two strands of DNA, right? Remember, there was like a ladder that's been twisted, right? So that's two strands of DNA. And this, it looks like one solid blue picture, or one solid blue ribbon in this picture. But as you know from the previous pictures, that's actually the phospholipid, good grief, the sugar phosphate backbone, right? So those are covalently bonded. But then what keeps the two strands together is this hydrogen bond. And as you know from chapter 2, and I told you this is going to be important again in chapter 10, hydrogen bonds are the weakest bonds and covalent bonds are the strongest bonds. So if you're tugging at this molecule, which is basically what happens on the cellular level, what's going to happen? These covalent bonds aren't going to break, right? Those ribbons are going to stay intact. What's going to happen is the whole strand's going to open up, right? The whole double helix is going to open up because those are weak bonds. Hydrogen bonds are weak. So it just nice and easily opens up. And then, like I said in the previous slide, the strand serves as a template. So I don't know if you guys can see it, but here's an A, right? So there's a machine that I'm going to tell you about later. I call it a machine, but technically it's a machine. It's a piece of the cellular machinery, so to speak. Anyway, it reads it and says, okay, there's an A. Now I need to bring in a T. So a T will come in here and attach. And then it reads this one, that's a C, right? So it says, okay, well, now I need to bring in a G. So a G will come in. I'm going to talk more about that as we move forward. But that's important right there, right? So that's basically how it works. We're going to get into more detail, but you need to understand this basic concept right here. The whole thing opens up, and those bases that are now exposed, that's the template. That shows you how to build the new strand. And there's a question on your study guide that might be on your exam that says something like, when cells go, or when DNA replicates itself, each new strand or each new double helix is made up of what? And the answer is each new double helix, right? Here's one new double helix. Here's another new double helix. Each new double helix is made up of the old original strand and a new strand. Because obviously the old original strand is the template. So that is a test, or that is a study guide question, and it might be a test question. Each new double helix of DNA consists of the old strand and the new template strand. Which, when you think about it, is crazy. Because then this thing is going to go on to duplicate itself later down the road. And then those two strands will go on to duplicate themselves. So somewhere, you have an original piece of DNA, like you have some really old DNA in you. Anyway, any questions about this slide? So remember, the new daughter strand, the old parental strand come together to make the new helix, the new double helix of DNA. If you download the PowerPoint, you can click on this little thing and watch a little animation that shows it happening. I'm also going to share some videos with you. When you watch those videos, and this one, if you see any words that you don't recognize, just ignore them. What's another good reason? Make sure you read your book, because you won't know. Like, you might be watching those videos like, oh, crap, I've got to memorize those words. But no, you don't. If they're not in the book, you don't need to memorize them, because we're giving you the Cliff Notes version in this class. There's a lot of parts that I'll show you up here that you don't learn about in the book. Oh yeah, that reminds me. Remember, Monday, if you bring in your textbook, you can get one-time extra credit, because I know some people don't have the textbook. So now's your time to get it. physically bring your textbook to class, and you'll get some extra credit for it. That's the one time I'm going to do this. Anyway, here's another little video you can watch. It's a lot less realistic. So this last one I just showed you is really realistic. It shows you what it might actually look like inside of a cell. This one's not as realistic looking, but it's more valuable as far as a teaching tool because it makes more sense to see how things work. Anyway, download the PowerPoint and watch it if you want. Same with this. This is a good example. So your book doesn't talk about template strand or lagging strand or primase. So again, read the book before you watch those videos, because if you see words on those videos that are not in your book, you can just ignore them. All right, here we go. So what does this? What opens up that strand of DNA that I just showed you? What reads the code and brings in the new bases, right? The thing is called DNA polymerase, and you need to know that for sure. That will 100% be a question on the exam. Probably the question will go something like, what enzyme is responsible for DNA replication? Because that's what we're talking about, right? And the answer is DNA polymerase. So let's talk about what it does. Obviously, it's responsible for DNA replication. That's what you need to know for the exam. But let's also talk about some other stuff here. In order to do the DNA replication, obviously, it has to make the bonds between the nucleotides of the new DNA strand. So like I just tried to show you earlier, like it reads that there's an A, so it says, okay, bring in a T. So it'll take that T and it'll bond it to the new strand that it's making. And then it'll bring in a new one and bond it to it. And I'll show you some pictures later that'll make a lot more sense. It does this by making sure that the base pairs match. So again, like I said, Wednesday, and you need to know this, and I'm going to draw it here again. A goes with T. That's horrible. And C goes with G. That's how it pairs up. So as the DNA polymerase is reading that, whenever it sees an A, it brings in a T. Whenever it sees a T, it brings in an A. Whenever it sees a C, it brings in a G. Whenever it sees a G, it brings in a C. Not that you need to know this. I'm going to put an X to it. But it is fast and it is accurate. How fast? It does about 50 nucleotides per second. So think about that. 1, 2, 3, right? So that's 150 nucleotides that just got built or attached when I did that. How accurate? Well, it only makes a mistake 1 in a billion bases. Which brings up a good independent work topic. How many bases are there in your genome? Because we know you have a bunch of A's and C's and T's and G's that make up your DNA. We know you have 23 pairs of chromosomes. But altogether, how many bases are there? Nice short independent work topic if you want to look it up. Also, look at this. Like I said, it's accurate, but I didn't say it's perfect. So yes, mistakes are made, and we'll talk about that. Without mistakes, we wouldn't be here. Without mistakes, which you're going to learn later are called mutations, we wouldn't have variation. And without variation, we wouldn't have evolution. And without evolution, we would just be a bunch of single-celled organisms and we would not exist. Another thing that DNA polymerase does, and I'm going to put an X through this as well, only indicating that I'm not going to test you on it, is it helps repair DNA damage. So not only does it build new DNA, but when your DNA gets damaged, then it goes in there and tries to fix it. So if you have high energy radiation from X-rays or UV light or whatever, anything that messes up your DNA, it tries to fix it. Doesn't always work, which is, again, one of the reasons you can get cancer from things like X-ray and UV light, because this is not a perfect process. Anyway, again, for the exam, these 3 top bullet points are the most important. DNA polymerase is responsible for DNA replication. Of course, these other 2, they basically just say how that happens. And I'm going to show you how that happens in future slides. Any questions so far? I'll go ahead and tell you this. There's going to be another polymerase on this exam. DNA polymerase is responsible for DNA replication. Any idea, any guesses what RNA polymerase might make? If DNA polymerase makes DNA, what do you think RNA polymerase makes? RNA, yes, and we'll learn about specifically what kind of RNA later, but I just wanted to go ahead and say that now because if it was just DNA polymerase in the exam, that'd be nice and easy. Actually, no, it's still easy. DNA polymerase makes DNA, RNA polymerase makes RNA. Someone's still going to mess that up on the exam, and I'm going to shed a little tear, and it's going to drip on my computer and break it, and then I'll get a new computer. It'll be great. Anyway, here's the picture again. So again, now that I've told you all that, try to picture an RNA, or I said it now, a DNA polymerase attached to this thing, and you can't see it doing its work, but it's opening this thing up, and it's reading this, and it's bringing in new bases, and it's attaching it to that new strand, and that's how it works. I'm going to show you some more pictures moving forward. But let's talk about it a little bit more. So we know DNA polymerase is the thing, that's the enzyme that does the work. But what's that process called? As you already know from chapter 8, it's called DNA replication, and it happens in the S phase of interphase. And I know it's been a while, so to remind you, the cell cycle, stupid thing, the cell cycle is made up of interphase and the mitotic phase. All right, there's the mitotic phase. Interphase is what most of it is, which is everything else. Good grief, I'm not even going to try. Anyway, interphase is a part, S phase is a part of interphase. S stands for synthesis, like DNA synthesis. Where does DNA replication happen? Like literally where on the DNA strand does it happen? It happens at a place called the origins of replication. I'm going to put a question mark through this, or next to it, indicating that I'm not sure if I'm going to ask you that on the exam or not. Maybe I will, maybe I won't. But yeah, that's where it happens. It happens at places called the origins of replication. And then it goes in both directions, as you can kind of see here. Like it starts here, starts here. There's multiple places on one strand of DNA where it starts, which makes sense. Because you don't want to, if you just start at one end and go to the other, that's going to take forever. Instead, it starts in multiple places and just kind of works its way out. So this is going to go working its way out, and this is going to work its way out. I'll show you some videos that really show it in action, and it makes a lot of sense. Some independent work topics, some other things you can look into. First of all, let me back up a step and talk about DNA polymerase. In this 100 level non-biology major course, we talk about DNA polymerase as if it's one enzyme. And as far as we're concerned, you can think of it like that. But if you want to dig deeper and look into it for independent work, you can look it up. It's actually, DNA polymerase is actually more than one enzyme. It's a team of enzymes working together and completely. When you put them all together, we call them DNA polymerase. But it's actually multiple things doing the job. Then the other question is, like I said, DNA polymerase is just bringing in those new bases. Like, all right, here's an A, let me attach it. Here's a T, let me attach it. Here's a C, let me attach it. Where are the bases coming from? Right? Where are we getting those bases? They're just floating around in your cell. So are your cells building each little base? Or is it when you eat something, does your body break down the DNA? Does it digest the DNA and then you have the nucleotides just floating around in your cells? Where do the cells come, or where do they come from? You can look that up for independent work. Also, the origins of replication. Like I said, all you need to know, if anything, is that this process starts on the origins of replication. But is there anything more to it? Is there like a certain code? Is this like a bunch of A's and then the DNA polymerase says, oh yeah, that's my code, that's where I attach, right? What is it about the origins of replication to let the DNA polymerase know that's where they need to attach? All stuff you can look up for independent work, but stuff you do not need to know for the exam. Any questions? Okay. Again, if you download the PowerPoint, you can watch this animation. This one I highly recommend because, like I said, that last picture I showed you, it's kind of hard to picture that bubble moving and those two bubbles moving outwards. But if you watch this little animation, you can see it in action. That one, let me see. Yeah, this one also shows it in action. Obviously, it's a little bit different. I don't remember how. It's been a while since I've seen them, but you can watch them if you want. And yet another one. Another thing you don't have to worry about. Again, like I said, lagging strand, leading strand, those are terms you don't need to worry about. 3', 5'. I don't think your book talks about that either. Okay, finally, we are now done with the first main bullet point, which brings us to the second main bullet point. Most of the exam is going to be based on this second main bullet point, which, as you can see, it's really long. This is probably the most important part of the chapter too, as far as your, I don't say everyday life is concerned, but the reason you're learning biology. Again, to truly understand things like vaccines and viruses or GMOs, to truly understand that stuff, you need to understand this. Hopefully, if you're paying attention, by the time I'm done with this, when someone tells you that the COVID-19 mRNA vaccine is gene therapy, you can laugh at them and say, oh no, that's not how that works. Because even at the 100 level non-biology major class or level, you'll be able to see that that's ridiculous. It doesn't work that way. So what I'm about to teach you is how we go from the code that's in your DNA, because like we've been saying this, your genes, which is what's in your DNA, are instructions on how to build proteins, right? I've been saying that for a long time. Now, finally, we're going to connect those dots and learn how it happens. Here we go. How the genotype makes the phenotype. As you already know, DNA provides instructions to a cell, right? Because sometimes your cells need enzymes to do their job. Sometimes DNA provides instructions to an entire organism. So the genotype is heritable information, which is the sequence of nucleotides and bases. And obviously, as you know, the phenotypes are the actions, or the phenotypes come from those proteins. So there's nothing really important here on this slide. This is all a review. Basically, what we're saying again is all that stuff you learned in the last chapter about heredity, it's DNA, right? It's DNA that's getting passed along. We said it's the genes and your genotype and the dominant allele versus the recessive allele. It's all right here. It's the DNA. That's all we're saying there. So then the question is, how does DNA provide these instructions? How is it that DNA says, all right, I need you to build this protein? How does that happen? That's what we're about to learn. For the next attendance word, I'm just going to underline it, won't even say it. That's the next attendance word. So if you're online, send a picture of that. Don't send the word, send a picture of that. Or a link, maybe even a link to a set of those. That one's a little bit tougher, right? Because usually you're not going to have one of those things laying around. You might, but you can get online and say, I need whatever. You can find something. Get creative. Anyway, there we go. How an organism's genotype determines its phenotype. We've already hinted at this, so a lot of this is going to be reviewed. But what is a phenotype? As you already know from last week, the phenotype is the physical traits. You already know that, right? The physical traits. The new part is from actions of a wide variety of proteins. I'm underlining this, yet you don't necessarily need to know it because there's not going to be a direct question about it. I'm not going to directly ask you that. So you already know that the phenotype would be like smooth chin versus dimpled chin, or blue eyes versus brown eyes, all that stuff you already learned, right? And I already said this, but now I'm officially saying it. How does all that stuff happen? It's because there's proteins dictating that, usually enzymes, right? So chin dimples, for example. What's your chin made of? Bone, right? That's not protein, that's bone. But there was proteins involved in the making of your chin, hence it's proteins that are responsible for your chin dimple or your lack of chin dimple. That's all this is saying. So even though a lot of these phenotypes have nothing directly to do with protein, they are still the results of enzymes. Your book gives some examples. So sometimes it is directly protein, like some structural proteins that help make up the body of an organism, right? So hair and fingernails, for example, that actually is protein. So yes, the gene directly is involved in that. But then again, like I've been hinting at, more importantly, not important for the exam, just important to understand how this works. Mostly it's enzymes, right? Enzymes are proteins. Enzymes catalyze almost every single reaction that's happening in your body. So a lot of your phenotypes are the results of chemical reactions, which are the results of enzymes. And again, for the exam, this is not that important. I just want to make sure you understand this. Because really, if you're not a thinker, we didn't even have to talk about this, and this wouldn't have even come up. But if you were a thinker, hopefully you would be thinking, like, how in the world, if genes are instructions on how to build proteins, but we're more than proteins, then how does that work? Now you know, right? It's enzymes mostly. So any questions about this slide? All right, there's no need to write any of this down yet, but you can if you want, I guess. This slide is an introduction to what you're about to dive into. So this is all just a scratching the surface, nothing to write down yet. DNA specifies protein synthesis in 2 stages. The first stage is called transcription. The second one is called translation. And again, I know I said you didn't have to write this down, and you don't yet. But my suggestion for studying this chapter is like I've suggested for other things. Memorize the big picture first, and then start adding details. So memorize this. If we had all the time in the world, and this was my only job, and your only class, and we could just take as much time as we wanted, I would just leave it at this. This would be the end of the lecture right here. I would say the DNA, the protein, is in 2 stages, transcription and translation. And I would let you guys marinate on that all night. And then we would come back tomorrow, and then I would get into more details. Because really, that's how it should work. So yes, first memorize that fact. First is transcription, then translation. Memorize that. And then once you have that memorized, then let's talk about what they are. And again, we're going to go really deep into this, but for now, let's just say transcription is going from DNA to RNA, and translation is going from RNA to protein, or polypeptide. For the sake of this conversation, this whole chapter, polypeptide is basically the same as a protein. To remind you the difference, because we haven't talked about it since chapter 3, a polypeptide is a chain of amino acids, right? You just add a bunch of amino acids together. That's a polypeptide. It's not technically considered a protein until it folds up and gets whatever shape it's supposed to be in. And the analogy I used back in chapter 3 was a piece of paper. That's just a piece of paper. And then if you fold it up into the right shape, then it is then a paper airplane, right? It's still a piece of paper when it's a paper airplane, but it's still technically at that point, it's called a paper airplane. It's like a polypeptide. It's just a polypeptide, just a string of amino acids until it folds up into its shape. Then it is considered a protein. It's still a polypeptide when it's a protein, but we consider it a full-blown protein once it's folded up into its shape. And that's the last time I'm going to talk about that for this chapter. I just wanted to remind you of the relationship between those 2 words. But again, for this chapter, as far as I'm concerned, those 2 words are interchangeable. We know that they're not, but for this conversation, they are. Because what we don't talk about in this 100-level non-biology major course is how we actually get from polypeptide to protein. We don't talk about how those things fold up. So another good independent work topic. Anyway, your book also points out that the relationship between genes and proteins is one of information flow. I don't really care about that. Your book pointed it out, so did I. I think your book pointed that out because back in chapter 1, they said one of the things you're going to learn a lot about in every chapter is information flow. So they're bringing it back. Like, hey, I told you you're going to learn about information flow. So yeah, there it is. But here we go. This bullet point, you already know it. I'm saying it again. The function of a DNA gene is to dictate the production of a polypeptide. And I've said this before too. I'll say it again. Don't memorize that verbatim. I'm not going to ask you that verbatim. Understand what that means. Right? A gene is a set of instructions on how to build a protein. However you want to word that to where it makes the most sense to you, memorize it that way. Don't memorize this word for word. I hate memorizing things word for word. That doesn't teach you anything. Someone can memorize that sentence and have no idea what it means, right? That's not helpful. You need to know that a gene, a DNA gene, is a set of instructions on how to build a protein or a polypeptide. Again, like I said, for this chapter, those two words can go hand in hand. They can be interchanged. So again, for the exam, the important thing here is to know that, again, a DNA, a gene, as a set of instructions on how to build a protein. How do we do that? Well, we go from DNA to RNA, and then we go from RNA to protein. And that is called, when we go from DNA to RNA, transcription. And when we go from RNA to protein, it's called translation. Big picture stuff. Like I said, memorize that first. And again, in a perfect world, I would end the lecture now. I'd give you a bunch of homework for this so you could reinforce that in your brain. And then once it was nice and set, then we'd come back and I would teach you the rest of it. But it's not a perfect world. But are there any questions about this slide before I go forward? All right, here we go. Before we get into it, we're going to do an overview. We're talking about how we go from nucleotides, which are DNA, right? How do we go from nucleotides to amino acids? Because remember, amino acids are the things that make up the protein. This is an overview. As I already said in the last slide or 2 slides ago, the information that's in your DNA is transcribed RNA, And then that information is then translated to the polypeptides, which is then folded into proteins. So again, this slide is nothing new. We just saw the same words in a different form 2 slides ago. Here's a picture of everything I just said. There's your DNA hanging out in the nucleus. We're going to transcribe that information into RNA. That RNA then leaves the nucleus and then is translated into protein. Again, big picture stuff. Memorize this before all the other stuff. Because you're about to learn this process. There's 3 stages to this process to make the RNA, and then there's something that has to happen before that RNA can leave. And then you're going to learn there's 3 stages to this process. And one of those 3 stages itself is broken down into 3 phases. It's so complicated. But this is nice and simple. So just remember that first. And also notice too, this is really big and important. Notice the way the arrows go. We go from DNA to RNA to protein, not the other way around. So when you get an mRNA vaccine, it's not gene therapy because the mRNA vaccines don't screw with your DNA. DNA makes mRNA. It's not the other way around. So your mRNA vaccines do not affect your DNA. It goes the other direction. Anyway, let's move forward. If you download the PowerPoint, you can watch another video to show what I'm about to teach you. All right. So we keep saying transcription and translation. And in some senses, you could hear those words outside of a biology class, right? So you transcribe something, meaning you write it. You translate something, meaning you go from one language to the next. It's the same situation here. In a sense, we're talking about a language. So what is the language of nucleic acids? Well, it's the sequence of the nucleotide bases. So that's sort of like saying, what's the language of English? Well, it's the sequence of the letters of the alphabet, right? The A's, B's, C's, D's, Z's, F, G's, right? You put those things together in different combinations to make words. Same here. We have the A's, mind you should have this memorized by now. But anyway, you have your A's, your T's, your C's, and your G's. You have 4 letters in your alphabet. But you just put those together in different combinations to make words, just like we do in English. Of course, we have more than 4 letters, but still, we put them together in different combinations to make words. Same thing here. And I'm going to put it next to this next bullet point because you don't need to know it, but it is a good independent work topic because you can see it says typical. It's very broad. The typical gene consists of thousands of nucleotides. Unlike in English, right, your typical word does not consist of thousands of letters, but in this language, a gene does consist of thousands. A typical gene does consist of thousands of nucleotides. So anyway, yeah, that's a good independent work topic. Specifically, how many nucleotides does a typical gene have? Thousands, that's a pretty broad term. Also, what's the smallest gene that we know about? How many nucleotides is the smallest gene that we know about? How many nucleotides is the largest gene that we know about? What organism has the largest genes? It might not even be humans. What organisms have the smallest genes? Why does Gene Belcher from Bob's Burger love music so much? Just kidding. You can look that up if you want, but don't send it as independent work. I already know the answer. It has nothing to do with biology. Anyway, DNA, as we already know from the previous slides, is transcribed to RNA. And the way this works is because the bases of the RNA are complementary to the DNA strand. In other words, it's a lot like DNA replication, where you open it up and you just read the code. The A's, the T's, the C's, and the G's. You read it and you say, all right, there's a C, I'm going to bring in a G. There's a G, I'm going to bring in a C, right? So it's the same, it's very similar to the way DNA replication works, which you already learned about. And we're going to talk a lot about this bullet point. So really, there's nothing to write down there yet because we're going to talk about it. Are there any questions so far? All right. Let's say the next word for attendance is acids. If you're online, send a picture of some kind of acid, whether it be LSD or hydrofluoric acid or a cartoon of some cartoon character pouring acid on some other acid. Another thing, if you're online, never mind. Be prepared. If you're online watching this, be prepared for the end. I'm going to have some We're calling audible, so be ready to think on your toes. That's all I'm going to say for now. Anyway, any questions about this slide? Okay, there we go. That was transcription that we talked about a little bit. Now we're talking about translation a little bit. And again, we're going to go into detail. So if you're taking notes, you can give your note writing hand a break if you want, because we're going to go into detail. But again, translation is going from RNA to proteins. But in the context of talking about the language of this process, what we're saying is if it's translation, obviously, you know, translation is going from one language to the other, right? So how many of you speak Spanish? A little bit, right? So if somebody came in speaking Spanish and then you guys don't speak Spanish and I translated it to you, that means we'd be going from one language to the other, right? Same here. Because when we go from RNA, when we go to DNA to RNA, yeah, we're changing things up a little bit, but it's the same language, right? That's still the nucleic acid language. When we go from RNA to polypeptides or proteins, now we're going to a different language. So that's where I would call it translation. So we're going from the nucleic acid language to the polypeptide language. Polypeptides, as you already know from chapter 3, are polymers. And as you already know from chapter 3, polymers are made up of monomers. What kind of monomers are these particular polymers made up of? And they're made up of amino acids. You do need to know that. I told you that in chapter 3. I told you it'd be coming back, and here it is. There are 20 amino acids. That's what makes up proteins. So again, if we're thinking about this in the language sense, to remind you that the DNA language is, that language has 4 letters. All of it, all the words in that language are made up of 4 letters. That's the nucleic acid language. Polypeptide language has 20 letters, so to speak, because they're not actually letters. Like they're literally letters when we're talking about nucleic acids, right? A's, C's, T's, G's, U's if we're talking about RNA. They're literally letters. In this case, they're not literally letters. They all have names, but it's still the same sense. If you're talking about a language, that's what it is. So it's the sequence of RNA bases that dictates the sequence of amino acids of the polypeptides. It really is translated. So the words that we have there'll be a word in the nucleic acid language made up of those letters. And that word says, all right, bring me this amino acid. And then the next word says, all right, bring me that other amino acid. And then they get attached. And then the next word says, bring me the other amino acid. That's literally how it works. You could almost think of this as like your chromosomes are books of instructions, right? Instructions on how to build proteins. And it's in words, right? It's literally written down. You read it and it says, this is how you build a protein. And then you take those words and then you physically build something. That's almost how this goes. So it'd be as if in this room, we had all the books on how to build stuff on this campus. And every time someone needed to build something, we need a bench outside of Wallace. Somebody would come in here, they would get the instructions, somebody would read it, it's written in English, and then that person could read it and then understand and build a bench. Because it would say, all right, you need 7 4x4s. All right, I'm going to give 7 4x4s. Attach them using this screw, right? And all those written words would then be translated into a 3-dimensional bench. Same thing here. You have written words in your nucleic acid language, and eventually that gets translated into this 3-dimensional protein. And that analogy will be coming back later. It's a good analogy, in my opinion. You'll see why. Here's a here's a visualization of what we just said. So here's a strand of DNA up top. It's got 3 genes on it. Obviously, there's going to be a lot more, but we're just focusing in randomly on this 3rd gene. We're going to zoom into that one little strand of gene 3. Obviously, we know a typical gene is thousands of letters long, so this is obviously just a small chunk of it. But yeah, here's the letters, right? The A's, the C's, the T's, and the G's. Those are the letters in the DNA. The RNA is then transcribed from that because it's complementary. So what goes with A, T goes with A, but remember, RNA doesn't have T, so wherever it should be a T, it's a U. So we have A, U, A, U, A, U. There's a C, so it brings in a G. There's a G, so it brings in a C, so on and so forth, right? It's transcribed. Just like if the construction workers came in here and said, hey, I need to build a bench outside of Wallace. I need the instructions, but I only have the 1 copy of instructions. So I'm not going to give them my whole book and say, yeah, just take the book with you. I'm going to write it down for them. All right, here's the instructions. I'm going to take what's there on the book. I'm going to write it by hand. I'm going to give it to them, and they can go translate that into a bench. Same here. When your cell needs to build something, it's not like your DNA just leaves your nucleus and it's done right there. No, we transcribe it. We write it down into RNA. And like I said, this is a language. The language, in this language, the alphabet has 4 letters, right? But they still build words just like we do in the English language. And as I'm gonna tell you this later, but I'm gonna go ahead and introduce you to it now. The words, each word in this language is made up of 3 letters. Right? They're all 3 letters long. And they're called codons. No need to write that down because I'm gonna officially give you that information later. This is still just an introduction. Anyway, any questions about this slide? Okay. Yeah, like I said, I already told you this, but now you can see it in writing. The flow of information from gene to protein is based on a triplet code. And I'm going to write down next to that, the triplet code equals word, right? So we already know the A's and the T's and the C's and the G's. Those are the letters. That's the alphabet of that language. But in this language, all the words are a triplet code. In other words, all the words are 3 letters long. Unlike the English language, right? You have one word like the word A, which is 1 letter long. Then you might have something like supercalifragilisticexpialidocious. I have no idea how many letters that is, but it's a lot, right? So we have a lot of variation in the English language. But in this language, all the words are 3 letters long. More importantly, for the exam anyway, we don't call them words. That'd be too easy. And then you guys would pass your exams too easily. We have to make it harder for you. And we can't call them words, we call them codons. So those 3-letter words are called codons. So when you hear me say codons, that's what I'm talking about. I'm talking about the words. If you hear me talk about bases, like the A's, the T's, the C's, the G's, and the U's, then I'm talking about the letters of that alphabet, of that language. When you hear me talk about codons, I'm talking about the words in that language. And again, all those words are always 3 letters long. And honestly, I mean, I'm going to ask you that, but now that I'm thinking about it, even that's a little bit not as important for you guys, for you non-biology major, 100 level course. Yeah, they're 3 letters long. And you're going to need to know that for the exam. But honestly, I think if you forget that after the semester's over, you'll be okay. As long as you understand generally how we go from DNA to RNA to protein, you're good. Because again, in your life, you're dealing with things like health issues, especially. This is good base knowledge to have. Anyway, these codons. In the DNA, as you already know, this bullet point is a review. I've already said it a few times. You've read it a few times today alone. The DNA, the codons of the DNA is transcribed into complementary codons in the RNA. So yeah, that's repeating what I've already said, except now we've just thrown in that word codons. Right? So previously I just said the DNA, the nucleotides from the DNA are transcribed into complementary nucleotides in the RNA. Right? That's what I said previously. Now I'm saying the codons in the DNA are transcribed into complementary codons of the RNA. And when I say complementary, to remind you, if the DNA says, I don't know, CCG, if that's what the word says, if those are the letters in the word, the codon, and the DNA, what do I mean by complementary? I mean, all right, well, if that's the DNA, then the RNA is going to be G, G, C, right? Because the G's go with C. That's all that is. There will be at least one question where I give you a strand of DNA. I say, transcribe this. Right? So I'm going to give you a bunch of A's and T's and C's and G's, and then the multiple choice question is going to give you a bunch of strands of A's and T's and C's and G's. And you have to pick the one that's complementary. And I already hinted at this on Wednesday, but if the question is asking you to transcribe something, that means your answer of the multiple choice exam is going to be RNA, right? So if the answer has a T in it, you can just go ahead and eliminate that one. Because if we're talking about transcription, you're making RNA. Right? You're going from DNA to RNA. So if your answer choices are RNA, and if they have a T in them, they're not correct. Because if it has a T, it's not RNA. Anyway, that's how we get from DNA to RNA. And again, that RNA codon is then translated into the amino acids that form the polypeptides. So these 2 bullet points are nothing new, except your book just threw in the word codons. We repeated what it said earlier, except now threw in the word codons. Any questions about this slide? Now, we already saw that picture. So again, we're talking about this word, which is made up of 3 letters, is transcribed into RNA with complementary 3 letters. That word, that 3-letter word, the codon, actually says, bring me a square amino acid. And this word says, bring me a circular amino acid. And this word says, bring me a triangle amino acid. And this word says, bring me another square amino acid. Of course, it's not, the amino acids aren't squares and circles and triangles, but your book's just trying to simplify it. That's a good place to stop. So the last word for attendance will be code. And here's the audible I'm calling. If you're online, I want you to send all those words, all those pictures by 8:52. So you've got 3 minutes to do it. So if you're not online and somebody's sending you a bunch of words, it's going to be a little bit harder for you. But if you've been doing this the whole time, you should have no problem sending me those pictures in 3 minutes. And of course, if you miss your deadline, you can just watch the video and answer the questions and get full credit. I recommend doing that anyway. All right, you guys have a good weekend.